So hello guys, welcome back with the new video. I'm sorry it took some time for the next video to come out. I have a lot of things planned. I had a lot of things planned which were going to be the following lectures, but due to some issues, due to some problems, things didn't go according to my planning before. Uh, Today we are going to study about the female reproductive system. I hope you have studied this topic before. We are going to just read along and we're going to study along. Don't worry if you have not read this. We will understand each and everything. I'm going to consider each student who has never studied this topic, never have been here. Don't worry, I will take care of the smallest thing. We will go into the smallest detail. This is not that big of a chapter, but it's very important for our basic understanding about the female reproductive system, and we will study histology of the female reproductive system. Uh, I know the video have been inconsistent because it was just not very motivating for not having views on the previous video, but never mind. We will try to be consistent. We'll try to help uh, as many people as we can. Even just a one person gets help through these videos, understands some kind of topic through this video, that is enough for me. So even if it's one person, I'm going to do that. So thank you so much for anyone who has watched my previous videos, and please keep supporting. Let's check the female reproductive system. So we have this chapter, female reproductive system. We're going to study about the ovaries, we're going to study about the uterine tubes, we're going to study about the major movements, the major events that take place in the fertilization. We'll study about uterus. We will see how the uterus thickening takes place when we have a menstrual cycle. We will see about the embryonic implantation after the fertilization. We'll see what decidua is. We'll see what the placenta is. We will also study about some organs like cervix, vagina. We'll see the external genitalia, and we are going to also study about the mammary glands, the breast development, because uh, it is somehow not the part of the female reproductive system, but somehow very dependent on the female reproductive system hormonally, and the process of lactation is very much in correlation with the process of fertilization and the female reproductive processes and events. So this part of mammary glands is also concluded. It's also included in the female reproductive system. So let's see what we have for the female reproductive system. Please let me know if my voice is okay. I just want to make sure if the voice looks fine. I think it should be fine. We'll see what decidua is. We'll see what the placenta is. Yes, looks perfect to me, and let's get into it. So yes, so the female reproductive system. Female reproductive system consists of the paired ovaries and oviduct. We have a pair of ovary in every female. We have an oviduct, also called as uterine tube. The same thing. We have the uterus, we have the vagina, and the external genitalia. Figure 22.1 has a complete detailed description of this, which is we have two set of ovaries here, then we have the uterine tube, we have the uterus, and then we have the cervix, we have the vagina, and we have the external orifice here. So this is all the female reproductive system. This is how the histology of the ovaries look in the figure C. Okay, let's get into more detail. This system provides the female gamete, oocytes. So female gametes are called oocytes. Provides the environment for fertilization and holds the embryo during its complete development through the fetal stage until birth. As with male gonads, the ovaries produce steroidal hormone and that control organs of the reproductive system and influence other organs. So these are basically the function of the female reproductive system. Beginning at menarche. This word menarche is important when the first menses occurs. The reproductive system undergoes monthly changes in structure and function, which are controlled by nephrohormonal mechanism. Nephrohormonal mechanism. Menopause is a variably timed period during which the cyclic changes becomes irregular and eventually disappear. In the postmenopausal period, the reproductive organs slowly involute. Although the mammary glands do not belong to the genital system, they are included here because they undergo changes directly connected to the functional state of the reproductive organs. So we have to remember here menarche and menopause. What is menarche? Menarche is basically the first menstrual cycle that a female experiences in the time of her puberty. And we need to know about menopause. Menopause is a period where the menses cycles becomes irregular and eventually disappears. This happens around 50 to 60 years of female's age, and this process is called menopause. Okay, so ovaries. Let's see what ovaries do and how dynamic their structure is. Ovaries are almond shaped bodies approximately 3 centimeter long, 1.5 centimeter wide, and 1 centimeter thick. Each ovary is covered by a simple cuboidal epithelium. The surface epithelium continues with the mesothelium and overlying a layer of dense connective tissue capsule. Okay, the tunica albuginea, like that of the testis, we have tunica albuginea also in the ovaries. Most of the ovary consists of cortex, a region where stroma of highly cellular connective tissue, and many ovarian follicles, varying greatly in size after menarche. So we need to know what we don't have exact separation of the stroma and medulla of the cortex and medulla in the ovaries, but we can observe the difference in the region. The region of cortex has a stroma which has lot of connective tissue and also consists of ovarian follicles. The most internal part of the ovary, the medulla, contains loose connective tissue and blood vessels entering the blood through the hilum from the mesenteries suspending the ovaries. There is no distinct border between the ovarian cortex and medulla. Early development of the ovary. In the first month of the embryonic life, let's read about the early development of the ovaries. In the first month of the embryonic life, a population of primordial germ cells migrate from the yolk sac to the gonadal primordia. So we need to remember that the female reproductive cycle starts even before this female has taken birth. When this female is in her fetal life, we see the occurrence of the primordial germ cell migrating from the yolk sac to the gonadal primordia. As the ovaries develop, these germ cells undergo synchronized mitotic divisions with incomplete cytokinesis. So these primordial germ cells start to divide, they go under a mitotic division, but they do not complete its mitotic division. They do complete all the phases, but they have an incomplete cytokinesis, producing a few million interconnected oogonia. We need to remember that oogonia are always interconnected because they did not undergo cytokinesis. This mitotic activity ceases at the 11 to 12th week gestation in human, and the clustered oogonia enter the long prophase of a first meiotic division. So then these oogonias and these clustered oogonias enter a meiotic division, and they are arrested in their prophase 1. Most of these cells undergo apoptic cell death, but others complete synapses and genetic recombination and then arrest without progressing to later stages of meiosis. These cells in meiotic arrest are called primary oocytes. So we need to remember that these cells that are in the meiotic arrest, the first meiotic arrest, they are called as primary oocytes. Now, not all oogonia are going to develop into a primary oocyte. A lot of them just go apoptic cell death. But the others who complete the synapses and genetic recombination, they go they are called primary oocytes, and each becomes surrounded by a flattened support cell called the follicular cell. So this oocyte is protected with a flattened layer of follicular cells to form a non-growing follicle. We understand. At birth, there are about 680,000 such follicles, sometimes called the ovarian reserve, of which about 460,000 remains at puberty. The others having been lost. As I said, the most of them go apoptotic cell death. They are lost through the degenerative process called atresia. So this loss of this oogonia is called atresia. This is a degenerative process. They degenerate, which only ends with menopause. So this degenerative process called atresia goes on not just at the fetal stage but throughout the life until the menopause of the female reproductive cycle. Because generally only one oocyte resumes meiosis, undergoes ovulation during each menstrual cycle, and the reproductive life of women is about 30 to 35 years. Only about 450 oocytes are liberated from the ovaries by ovulation. Altogether, all others degenerate through atresia. So we need to know that a woman has a reproductive life of 30 to 35 years. If we calculate a 28-day cycle, a reproductive cycle of 28 days where one ovulation will occur and one oocyte will be produced in 28 days. With this calculation, we can estimate that 450 oocytes throughout the year is what ovulated by a female reproductive system. Rest all of them undergo an atresia. Okay. What are ovarian follicles? We know of primary follicle now. We have a primary oocyte surrounded with a flattened follicular cells. That is a follicle. But let's see what ovarian follicles, how what happens to them later and how they grow and what events do they follow? An ovarian follicle consists of an oocyte surrounded by one or more layers of epithelial cells called the basal lamina. Like basic thing, consists of oocyte surrounded by one or more layer of epithelial cells within a basal lamina. Now the follicles that are formed during fetal life, that is, we know in fetal life we have a primordial follicle consists of a primary oocyte enveloped by a single layer of flattened follicular cells. This is a follicle, primordial follicle. These follicles occur in a superficial ovarian cortex. They appear in where? The location is ovarian cortex. The oocyte is the oocyte in the primordial follicle is spherical and about 25 micrometers in diameter, with a large nucleus containing chromosomes in first meiotic prophase. So, with a large nucleus containing chromosomes in the first meiotic prophase, the organelles tend to be concentrated near the nucleus and include numerous mitochondria, several Golgi complexes, and extensive rough endoplasmic reticulum. The basal lamina surrounding the follicular cells mark a clear boundary between the follicles and the vascularized stroma and act as a blood follicle barrier. So, this basal lamina that is surrounded between the follicle, then it marks as the blood follicle barrier. This is the blood follicle barrier. Okay. Follicular growth and development. We are going to see a lot of diagrams here because that is required for a follicular growth and development to understand how the follicle grows. Beginning in the puberty with the release of the FSH, follicle stimulating hormone FSH, from the pituitary, a small group of primordial follicles each month begin a process called follicular growth. We need to know that now there is a conflict between the textbooks. Some textbooks say that primordial follicle to the primary follicle is not a function of FSH, but we're going to follow what is here in the text for exams. Beginning in the puberty with the release of FSH, which is a follicle stimulating hormone secreted by the pituitary, a small groups of primordial follicles each month begin a process of follicular growth. This involves growth of the oocyte, This also involves the growth of the proliferation and changes in the follicular cells, as well as the proliferation and differentiation of the stromal fibroblast around each follicle. We need to have changes inside the follicle and also around the stromal environment in the cortex. So, selection of the primordial follicle that undergoes growth and recruitment early in each cycle, and the dominant follicle destined to ovulate that month both involve complex hormonal balances and subtle differences among follicle in FSH receptor numbers, aromatase activity, estrogen synthesis, and other variables. So these are the variables: various receptors number, aromatase activity, estrogen synthesis capability of this. These are basically one of the factors that variables and drives the selection process. Promoted by FSH and oocyte grows. It grows rapidly during the first part of the follicular development, reaching a diameter of about 120 micrometers. Oocyte differentiation includes the following. Now, what does basically when we say an oocyte differentiation? We know the growth of, we know what is the meaning of growth of oocyte. But let's now see what is the differentiation processes. So, growth of the cell and nuclear enlargement. This is what takes place. In this process, also mitochondria becomes more numerous and uniformly distributed because we are going to require a lot of energy for this process. Rough endoplasmic reticulum becomes much more extensive when Golgi complexes enlarge and moving peripherally. So we, we are going to need more energy to create more protein. So we have more rough endoplasmic reticulum, and we also need to package this lot of protein, so we need Golgi as well. Sorry. Formation of specialized secretory granules called the cortical granules containing various proteases. These lie just inside the oocyte's plasma membrane and undergo exocytosis early in the filtration, early in the fertilization. My bad. Sorry, fertilization. So we need to know now this increased mitochondria, increased RER, and the big size Golgi. What are they going to do? They're going to produce these granules, and these granules are going to contain proteases, and these granules are called cortical granules. uh, they lie inside the oocyte's plasma membrane and undergo exocytosis early in the fertilization. We will know when we will study about the events of fertilization that why these granules are important and why did in the earliest stage of this follicular growth we created these granules. We'll see. Now, follicular cells undergo mitosis and form a simple cuboidal epithelium. These follicular cells grow around the oocyte and they get multilayered. The follicle is now called a unilamellar primary follicle. Right now, it's still a single layer but a thick, and it's cuboidal. The follicular cells continues to proliferate, means grow, forming a stratified follicular epithelium, the granulosa. Now, this follicular cells grow, and this follicular epithelium is called granulosa, and the cells of this epithelium are called granulosa cells, in which the cells communicate through gap junctions. This is important. How does these cell communicate? Gap junctions. Follicular cells are now termed granulosa cells, and the follicle is multilaminary follicle. So we need to know when we have a unilamellar follicle, we are going to call it a simple cuboidal follicular cells, which has a simple cuboidal epithelium. But when we call them when they are get stratified, when they are stratified follicular epithelium, we call those granulosa cells, and they communicate through gap junctions. And this situation of the follicular cells, the granulosa cells, is called the multilamellar but primary follicle. Still avascular, we have not given any vasculature yet, and surrounded by a basement membrane. Now, between the oocyte and the first layer of the granulosa cells of the growing primary follicle, extracellular material accumulates at the zona pellucida. First time here we have conversation of zona pellucida. In the unilamellar cell, in this figure 22.3, we can see that in the unilamellar primary follicle, we have started formation of zona pellucida, this small membrane, thin membrane here around the follicular cells here. But now we have a zona pellucida properly developed in the multilamellar primary follicle. Okay, 5 to 10 micrometers thick and containing 4 glycoproteins secreted by the oocyte. This is what zona pellucida is. It's quite thick. It's 5 to 10 micrometers thick, and it consists glycoproteins that are secreted by oocyte. Anything now that these layers will consist of, that will be secreted by oocyte. Who else will do that? But most important secretions and most important contents of this zona pellucida are the ZP3 and ZP4 components. The zona pellucida components ZP3 and ZP4 are important sperm receptors binding specific proteins on the sperm surface and including acrosomal activation. Philopodia of granulosa cells and microvilli of the oocyte pellucida, allowing communications between these cells via gap junctions. Again, we have a repetition of the communication between the cells takes place through gap junctions. Stromal cells immediately outside each growing follicle. We now see have the follicles are growing, and the stromal cells in the cortex around the follicle also needs to change themselves. So stromal cell immediately outside each growing primary follicle differentiate to form the vascularized follicular theca, which subsequently differentiates further as two distinct tissue around the follicle. We have this theca cells from the environment in the stroma of the cortex, and these cells later turn two different layers, the inner theca and external theca. Let's see what is their significance. A well vascularized endocrine tissue, it's an endocrine tissue, so it secretes hormone, the theca interna, with typical steroid producing cell secreting androstenedione, which is basically androgen. This precursor molecule diffuses into the follicle through the basement membrane, and in the granulosa cells, the enzyme aromatase converts it to the estradiol, an FSH dependent function. This function is dependent on FSH also, guys. So we have this FSH activity required, FSH presence required, starting from the primordial follicle till we also have a theca interna here, and we have already a multilamellar follicle. This estrogen, the estradiol, basically estrogen, this estrogen returns to the theca and stroma around the follicle, enters the capillaries, and is distributed throughout the body, inducing changes characteristic of the puberty. So remember, we this theca interna produces androgen, which this androgen travels to the granulosa cells. This granulosa cell converts the enzyme aromatase converts this androgen, the androstenedione, to what? Estradiol. And this estradiol, basically estrogen, then goes through theca's vasculature throughout the body. This is all the feature of theca interna. Let's see what theca externa does. A more fibrous theca externa with fibroblasts and smooth muscle merges gradually with the surrounding stroma. As the primary follicle grows, they move deeper in the ovarian cortex. Within such follicles, small spaces appear between the granulosa layers as the cells secrete follicular fluid. This fluid accumulates the spaces. Who secretes the follicular fluids? Who secretes this antrum? Granulosa cells. This fluid accumulates the spaces, enlarge, and gradually coalesce, and the granulosa cells reorganize themselves around a large cavity called the antrum, producing follicles now called secondary or antral follicle. We need to know that when we have an antrum, the fluid, that is the follicular fluid that is secreted by the granulosa cells, when we have the presence of this antrum, we call this follicle as the antral follicle or a secondary follicle. Now follicular fluid contains the large gag hyaluronic hyaluronic acid, growth factors, plasminogen, fibrinogen, and anticoagulant heparin, sulfate proteoglycan, and high concentration of steroids with binding protein. So this is very important. These factors and what does the antrum contains. As the antrum develops, the granulosa cells around the oocyte from a small hillock, the cumulus orifice, which protrudes into the antrum. We'll check the figure for figure 22.322.7b for what this cumulus orifice is, because this granulosa cell, they grow and they create this hillock, and this hillock is called the cumulus orifice. 22.3. Yes, so this part here is cumulus orifice. So this part, how important is it? Because it is just next to the oocyte, and it is going to play a very important part for ovulation. Let's see how and what it does this do. Which protrudes into the antrum. It grows and it protrudes into the antrum, and the tightly adhering granulosa cell immediately surrounding the zona pellucida make up the corona radiata and accompany the oocyte when it's leaving the ovary of the ovulation. So these are basically granulosa cells around the oocyte. They get compact in a layer called corona radiata because they are ready to move out from this rupture at the place of cumulus orifice, go out in the process of ovulation with the oocyte. And these granulosa cells that go out with the oocyte are basically a layer of corona radiata around it. So this single large antrum now, which has the corona radiata looking differently, which also has cumulus orifice around it, around of cumulus orifice of the granulosa cells that is protruded in the antrum. When these things become visible, evident, then we call this follicle a mature follicle or a Graafian follicle. Now, why Graafian follicle? We all know a lot of scientists who discover things would like to make their name big. So, named this Graafian follicle is named after the 17th century reproductive biologist Regnier de Graaf. Graaf for Graafian, thus a Graafian follicle. Now, this Graafian follicle, the large mature follicle, rapidly accumulates more follicular fluid and expand to a diameter of 2 centimeter. 2 centimeter is quite huge, and a mature follicle forms a bulge at, remember where we discussed about 5 to 10 micrometers of diameter of the primordial follicle to the mature follicle now, which is 2 centimeters. A mature follicle forms a bulge at the ovary surface, visible with ultrasound imaging. The granulosa cell layer becomes thinner at this stage because its cells do not multiply in the proportion to the growth of the antrum. The antrum is keep growing, keep growing. A mature follicle has thick thecal layers and normally develop from a primordial follicle over a period of about 90 days. So all this process from the primordial follicle up till this ovulation, it all took 90 days to create one mature follicle. Okay, let's check if my voice is going perfect. So let's see what we have. To the antrum, it grows and it protrudes into the. Yes, looks perfect to me. So this is all the development of the follicle and the growth, and we have a mature follicle that is now ready for ovulation. Let's see how it the dynamic these events are. But we also read something about atresia. This one primary follicle worked really hard and survived and made it to become a mature follicle. But let's see what happens with the others in these 90 days period. What happened to these? Unfortunately they undergo a degenerative process called atresia. So most ovarian follicle undergo the degenerative process called atresia, in which follicular cells and oocytes undergo apoptosis and removal by phagocytotic cells. Follicles at any stage of the development, including nearly mature follicle, may become atretic. So we need to know that maybe they come close to become mature but still had to undergo atresia. You never know, because complete mature will be just one, because only one oocyte will get ovulation. So people, some follicles might be in the secondary stage and they get get atretic. They are in primary stage, multilamellar, unilamellar, and get atretic. We never know. Atresia involves detachment of the apoptic granulosa cells. They get detached, the granulosa cells, autolysis of the oocyte, and collapse of the zona pellucida. Macrophages invade the degenerating follicles and phagocytose. So these macrophages are basically from the vasculature from the medulla. Phagocytose the apoptic material and other debris. Typically, during a woman's fourth decade, menopause begins when atresia and ovulation have reduced the ovarian reserve to fewer than 1,000 follicles. You remember 460 lakh follicles we had? First, we had 600,000 follicles. Then it came to 460 follicles. And now, at the end of just 1,000 follicles, we observed menopause. During a typical menstrual cycle, one follicle becomes dominant and develops farther than the other. The dominant follicle usually reaches the most developed stage of follicular growth and undergoes ovulation, while the other primary and antral follicle undergo atresia. So, although sorry, although their oocytes are never directly used, the large growing follicles each month produce much estrogen before becoming atretic. As described later, this estrogen stimulates preparation of the reproductive tract to transport and sustain the embryo if the oocyte from the dominant follicle is fertilized. Of course, let's see what happens next. We have now ovulation and its hormonal regulation. We'll see, now we have a mature follicle that is ready for getting ovulated, and this is around the 14th day of the cycle. We'll study the menstrual cycle complete, breaking down into day to day enhancements, but 14th day is what ovulation takes place. Let's see what ovulation is. Just check what we have. We have corpus luteum to study, and then we go to uterine tubes. So this is what ovulation is. Ovulation is the hormone stimulated process by which oocyte is released from the ovary. Ovulation normally occurs midway through the menstrual cycle, that is around the 14th day of a typical 28-day cycle. In the hours before ovulation, the mature dominant follicle bulging against the tunica albuginea develops a whitish or translucent ischemic area, the stigma, in which tissue compaction has blocked blood flow. So what happens that we have a layer of tunica albuginea, and we have this ovary, the part of this ovary growing, and this connection with the tunica albuginea here creates an avascular area, a ischemic area, a translucent ischemic area. This is called stigma, and eventually that is going to be the area from which where the ovulation is going to take place. Uh, in humans, usually only one oocyte is liberated during each cycle, but sometimes either no oocyte or two or more stimulate simultaneously oocytes may be expelled. This is an exception, but mostly only one oocyte is going to be expelled. Just before ovulation, the oocyte completes the first meiotic division, which it began and arrested in prophase during fetal life. The chromosomes are equally divided between the two daughter cells, but one of these retain almost all of the cytoplasm. That the cell is now secondary oocyte, and the others become the first polar body. So now, when the chromosomes are equally divided, this oocyte goes completes its first meiotic division, we have two products. One is now a secondary oocyte and one is the first polar body. Now, chromosomally, we need to understand that they divide equally because it's normal meiotic division, the first meiotic division, and we have chromosomes equally divided. But cytoplasm plays an important role here. Most of the cytoplasm travels with one set of this chromosome, and that eventually becomes our secondary oocyte, the main oocyte that is going to transfer the genetic material further. And the one with good, equally genetically strong but no cytoplasm, we call it the first polar body, which is later going to get degenerate, but it's first polar body, a very small non-viable cell containing a nucleus and a minimal amount of cytoplasm. Now, immediately after expulsion of the first polar body, the nucleus of the oocyte begins the second meiotic division, but arrests at metaphase. Now we remember that we arrested in the prophase when we were doing the first meiotic division, and it started in the in when did it start? It started in the embryonic life of this female, but it did complete its first meiotic division when it is now ready for ovulation. And we have this started second meiotic division and it gets arrested in the metaphase. Let's see what happens. Immediately after the expulsion of the first polar body, the nucleus of the oocyte begins the second meiotic division but gets arrested at the metaphase and never completes meiosis unless fertilization occurs. So we know that second meiotic division gets never completed if we don't have fertilization. Only in the presence of sperm we will undergo the next division. Let's see what happens. Very interesting. Okay, so as mentioned before, follicular development depends on FSH. We know that all the FSH, all the follicular development from the primordial follicle to the mature follicle, everything was dependent on FSH from the pituitary gonadotropes, whose secretion is stimulated by gonadotropin releasing hormone GnRH from the hypothalamus. We know from the basic endocrinology that every hormone that is secreted by pituitary gland gets a signal to get secreted from the thalamus by GnRH, gonadotropin releasing hormone. Okay, so figure 2211 summarizes the main hormonal interaction that regulates follicular growth as well as the ovulation and formation of the corpus luteum. We'll study about the hormonal things that happen. Just this one diagram and you will understand everything, what hormonal changes take place. So let's read. Okay, note that negative feedback of estrogen and progesterone on the hypothalamus and anterior pituitary is reinforced by polypeptide hormone inhibin, also produced by granulosa and luteal cells. In the days preceding ovulation, the dominant follicle secretes higher levels of estrogen that stimulate more rapid pulsatile release of GnRH from the hypothalamus. The increase we need to know about inhibin. Inhibin is important for the negative feedback, because GnRH was the one that gave a positive feedback, stimulated the pituitary, and we have secreted FSH. Later in the cells we secreted androgen that was later converted into estrogen. But for this negative feedback of uh, estrogen and progesterone, we have inhibin. Dominant follicles secrete higher level of estrogen that stimulates more rapid pulsatile release of GnRH from the hypothalamus. The increased level of GnRH causes a surge of LH released from the pituitary gland and rapidly triggers a sequence of major events in and around the dominant follicle. So what happens? What is this LH surge? So just before the ovulation is going to take place, there is a surge of LH increasing. Now, this is a part of research that should go negative feedback. We already have an FSH that is working, but why We also have a slight increase in FSH at this moment, but we have the major increase of LH, the luteinizing hormone. And it's a part of research that why does such a high spike of LH is observed, but we know that it is observed just before the ovulation. I guess the nature doesn't want anything to fail, and it wants LH to be ready. It wants everything hormonally perfect for nothing to fail, and that's why we have an LH surge, very dynamic. But what major events take place around this LH surge? Why this LH surge? So meiosis one is completed by the primary oocyte. As I said, just before the ovulation in the mature follicle, our oocyte will complete its first meiotic division, and second meiotic division never gets completed if there's no fertilization. And if there is a fertilization, we'll see what happens. Yeah. So meiosis one is completed by the primary oocyte yielding a secondary oocyte and the first polar body that degenerates. Granulosa cells are stimulated to produce more follicular fluid containing prostaglandins, proteoglycans, and proteases that remove the surrounding blood follicle barrier. The granulosa cells in the cumulus orifice uh, the granulosa cells in the cumulus operus oocyte complex mainly release hyaluronin, which increase the fluid viscosity and swells to greatly increase the extracellular volume within this complex, loosening the outer cell and dissociating it to form the follicle wall, ballooning at the stigma. The ovarian wall weakness Okay, the ovarian wall weakens as activated plasminogen plasmin from the broken capillaries degrade collagen in the tunica albuginea and surface epithelium. And fourth, smooth muscle contraction begin in the theca externa triggered by prostaglandins diffusing from the follicular fluid. So all these things, the hyaluronin and the antrum and this ballooning of the stigma, the balloon, the uh, that is pushed around the tunica albuginea, the ovarian surface, the ovarian wall get weakens, for the oocyte to move out and protrude, and this plasmin, we remember, that was secreted by the granulosa cells in the antrum, is going to help. And we have the smooth muscles that are present in ovaries to undergo contraction, which eventually everything leads up for this ovulation day. LH surge, very dynamic, very interesting. The increasing pressure with the follicle and weakening of the wall lead to rupture of the ovarian surface of the stigma. The oocyte and its surrounding corona radiata, the oocyte and the corona radiata, along with follicular fluid, are expelled. The follicular fluid, the antral fluid, are expelled by the local smooth muscle contractions. The ovulated secondary oocyte, we know that this oocyte is a secondary oocyte, adheres loosely to the ovary surface in the viscous follicular fluid, and as described later, is drawn into the opening of the uterine tube where fertilization may occur. If not fertilized within about 24 hours, the secondary oocyte begins to degenerate. Cells of the ovulated follicle that remain in the ovary redifferentiate under the influence of LH and give rise to corpus luteum. Now, after the ovulation, we have the ovaries secreted. We'll see where the ovary, sorry, we after the ovulation, we have this oocyte secreted. We'll see where the oocyte goes, but all the rest part left behind is the theca interna, externa, and the other left material from the follicle gives rise to corpus luteum. Let's see what this corpus luteum does. It's not done. Corpus luteum is going to be very helpful after ovulation. Now the ovulation is done. The granulosa cells and the theca interna of the ovulated follicle reorganize. Some granulosa cells and the theca interna of the ovulated follicle reorganize to form a large temporary endocrine gland. Corpus luteum is an endocrine gland. It's going to secrete hormones. Is corpus luteum. in the ovarian cortex. Ovulation is followed immediately by the collapse and folding of the granulosa and thicker layers of the follicle wall, and blood from the disrupted capillaries typically accumulates as a clot in former antrum. It's accumulate, the waste part, the granulosa cells, they get folded in, and accumulated in the former antrum. The granulosa is now invaded by capillaries. It's not those granulosa cells before were avascular, but in the corpus luteum they are vascular, they are invaded by capillaries. This is very important. Cells of both the granulosa cells and theca interna, theca always was vascularized, theca never was avascularized, and now those vasculature has came down to granulosa cells as well. uh, Change both cells of both the granulosa and the theca interna change histologically and functionally under the influence of LH. We already after the LH surge we have this LH influence all over. We don't need FSH anymore. The ovulation is done, the follicular growth is done, and we don't need FSH anymore. Becoming specialized for more extensive production of progesterone in addition to estrogen. So estrogen was getting produced already by, you remember in the production of estrogen also two layers were important. The two cells, theca interna and granulosa cells. Theca interna created androgen, which traveled to the granulosa cells, and granulosa cells converted it to estradiol, that is basically estrogen. And this estrogen is being produced already, but to the addition of estrogen now, this corpus luteum is also going to produce progesterone. Granulosa cells increase greatly in size without dividing and eventually comprise about 80% of the corpus luteum. They are now called granulosa lutein cells and have lost many features of protein secreting cells to expand their role in aromatase conversion of androstenediol into estradiol, as I said. The former, the before theca interna forms the rest of the corpus luteum as theca lutein cells. We cannot just, because the structure has changed, we cannot just call them granulosa cells and theca interna. We are going to call the same structures in the corpus luteum as granulosa lutein cells and theca lutein cells. These cells are half the size of the granulosa lutein cells and are typically arranged aggregated in the folds of the wall of the corpus luteum, which, like all endocrine glands, become well vascularized. Luteinizing hormone causes these cells to produce large amount of progesterone as well as androstenedione. The short-term fate of the corpus luteum depends on whether a pregnancy occurs. The ovulatory LH surge causes the corpus luteum to secrete progesterone for 10 to 12 days. Without further LH stimulation and in the absence of pregnancy, both major cell types of the corpus luteum cease steroid production and undergo apoptosis. The, the regression of the tissue, a consequence of the decreased secretion of progesterone, is menstruation. This is what menstruation is at the after the just after the LH surge. This, the consequence of decreased secretion of progesterone, because progesterone is required to keep the endometrium thickened, but as the progesterone is decreased, endometrium is not going to be thickened and it starts shredding. We'll study each day, and each day of the menstrual cycle and what changes take place. uh, I think I'll need to stop this video and get starting the new one, because we have crossed a limit, I think, for yes, 50 minutes. We are done 50 minutes. So uh, wait for the next lecture, guys, and we'll continue just from this point here, and we'll continue this exact topic in the next video. Thank you.